Hello and welcome back to Elo's Notes. In today's video, we will explore the stages of mitosis. Before we start with the mitotic phase, let's quickly review the cell cycle. Remember that the cell spends almost 90% of its life in interphase, which includes G1, S, and G2. Now, although the cell is not dividing yet in G2, but towards the end of G2, the cell starts getting ready for cell division. For example, towards the end of G2, the centrosomes duplicate. They don't migrate just yet, they stay near the nucleus, but now we have two centrosomes. The major phases of mitosis are prophase, metaphase, anaphase, and telophase. You will also see prometaphase or late prophase. And usually we attribute the events that take place here to prophase if we don't want to get too technical about it. But if you're taking a higher level biology, then we have to distinguish between the events that take place in prophase and prometaphase. An easy way to remember the order is pick me at 10. So pick me at 10. Prophase, metaphase, anaphase, telophase. In fact, this is something my biology teacher back when I was in high school taught us. It is very important to understand that mitosis is the division of the nucleus into two identical nuclei. And once those two identical nuclei are formed, the cell itself starts to divide in a process known as cytokinesis. Let's start with P prophase. The centrosomes migrate to opposite ends of the cell. And what helps them do so is the growing microtubules that we refer to as spindle fibers. The centrosomes also form shorter microtubules that are called asters. Why? Because they look like stars. So together, the centrosomes, the asters, and the microtubules form what we call the mitotic spindle. Next, if we direct our attention to the chromatin, we notice how they condense and coil into chromosomes that become clearly visible under the microscope. In prometaphase or late prophase, the nuclear envelope breaks down. Why? Because if we don't have a nucleus anymore, then the microtubules and the chromosomes can move more freely in the nuclear region. And remember how chromatin already condensed into chromosomes, but here they become even more condensed and they get attached to the spindle fibers, to the microtubules through the kinetochores. The kinetochores are protein structures at the centromeres. Before we move on, let's talk about chromosomes. Remember that chromosomes exist in pairs, a maternal set and a paternal set. So you see you have two sets here, the blue ones and the pink ones, a maternal set and a paternal set. In the S phase of interphase, DNA replication takes place, which means now we have an extra copy of every single chromosome. The two identical copies of the same chromosome. They're called Sisterchromatids. Why? Because these are exact copy-paste of each other. They contain the exact same genes. However, the two chromosomes in a pair are called homologous chromosomes. Why? Homo means similar. They contain similar genes at the same locations. However, they are different because one is of maternal origin and the other is of paternal origin, and hence they carry different alleles. The sister chromatids join at the centromere, and this is where the kinetochores form. So now let's move on to metaphase. Meta, let's meet in the middle. This is a nice mnemonic for you to remember that in metaphase, all the chromosomes meet at the center of the cell. So as the microtubules keep growing, they move the chromosomes to the center of the cell. They create this imaginary center or plate, which is called the metaphase plate. In anaphase Sisterchromatids separate. It's the sisterchromatids, not the homologous chromosomes, because homologous chromosomes are not identical chromosomes. And here in mitosis, we want to end up with two nuclei that are identical. And that's why it's the sisterchromatids that separate. They begin moving towards the opposite ends of the cell. This is made possible because the microtubules with kinetochores, meaning the ones that have chromosomes attached, they shorten. And the non-kinetochore microtubules, which means the ones that don't have attached chromosomes, they elongate. Okay? So some are elongating, some are shortening. What does this create? This movement not only separates the chromatids, but it also elongates the cell. Finally, the last stage is telophase. In this phase, everything we mentioned in prophase will be reversed, meaning the chromosomes loosen up. They become chromatin again, and we can't see them again under the microscope. The nuclear envelopes, they reappear. So mitosis ends here. What will happen next, which is the actual splitting of the cell into two, is cytokinesis. In animals, a cleavage furrow, which is a dent that appears and it starts to go deeper and deeper until the cell pinches into two. This is it for today. I hope you found it easy and useful. Don't forget to like this video and leave comments, and I'll see you in the next one. Bye!